2020 in Genoa to present this plenary session on the Ubiquitin-Proteasome system and primary immunodeficiencies. And I'm just sorry that it's not in person and that we have to do this remotely. So we have three wonderful speakers. The first is Ivona Aksentijevich, who is one of the queens of autoinflammation working at the NIH. And then we have Fabio Martinon, who works in Lausanne, who's one of the kings of the inflammasome and now of the proteasome. And then we have a princess of pomp, Cecilia Poli, who is now working in Chile, but previously worked with Jordan Orange in Baylor. And I look forward very much to this session, which will be followed by a question and answers session at the end. Thank you.
Hello to everyone and welcome to this session. First, I would like to thank the organizers for their invitation to participate in this exciting session, and I wish all of you to enjoy the upcoming talks. So what I would like to do today is to review for you the novel genetic causes of autoinflammatory diseases which are caused by dysregulation in the ubiquitin-proteasome pathway, and to discuss the cell-specific and transcript isoform-specific functions of various UPS-related proteins in the regulation of inflammatory signaling. Before I start my presentation, I would like to recognize the three scientists who were jointly awarded the Nobel Prize for their work on the discovery of ubiquitin-mediated protein degradation, and I like these two quotes they used to describe their discovery. So, basically, ubiquitin-mediated protein degradation pathway is essential for regulation of all cellular processes, including immune and inflammatory responses. The ubiquitination it's a type of post-translational modification, reversible protein modifications that involves the attachment of these highly conserved ubiquitin proteins, either in the form of monomers or polymeric ubiquitin chains. Based on the type of linkage between ubiquitin monomers, there are different types of chains. The best known are K11, K48, K63, and also MET1 or linear ubiquitin chains. Because this process is reversible, it means that essentially these ubiquitin proteins will have to be removed from its substrate, and this process is known as deubiquitination, and it's mediated by deubiquitinating enzymes, also known as DAPs. Many of these DAPs have cell-specific functions. Ubiquitination involves the covalent attachment of ubiquitin proteins to its target substrate by the coordinated action of ubiquitin activating enzyme, also known as E1, ubiquitin conjugating or transferring enzyme, known as E2, and ubiquitin ligase enzyme or E3 enzyme, which finally results in the attachment of ubiquitin proteins to its substrate. What you can see from this slide is that there is a big difference in the number of E3 ligases versus E1 enzymes. That basically suggests that E3 ligases have substrate and also cell-specific functions. However, what is surprising is that there are only two isoforms of E1 enzyme, which is essential to initiate this whole process. Moreover, one of these enzymes resides on X chromosome. So you would think that, considering the important role of this protein, that there should be more redundancy for this protein in or gene in human genome. The disease that I will discuss later on in my talk actually is disease which is caused by somatic mutations in the major isoform of E1 enzyme, which also suggests that germline mutations in this enzyme are likely incompatible with life. The type of ubiquitination determines the fate of the modified proteins. So proteins that are modified with or conjugated with K48 ubiquitin chains are sent for proteasomal degradation, while attachment of these K63 or linear ubiquitin chains creates activation signal in these cells. As I already mentioned, the ubiquitination process is negatively regulated by the ubiquitinating enzymes or DAPs, and there are more than 100 of them, and many of them have cell-specific functions. Going back to monogenic autoinflammatory diseases, basically they are inherited diseases of the innate immunity, and they result from antigen-independent activation of the innate immune pathways. These patients present with seemingly unprovoked excessive inflammation, which is predominantly mediated by myeloid cells. This is in contrast to patients with more common and better known autoimmune diseases who basically have, in whom inflammation is mediated by the cells of the adaptive immune system. So these patients have high titer autoantibodies or antigen-specific T cells. So this term was introduced to distinguish patients with autoinflammatory from patients with autoimmune diseases, and that was indeed true for the prototypic autoinflammatory diseases such as familial Mediterranean fever. However, as the boundaries between the innate immunity and adaptive immunity are not clear, there are many patients that present with the features of autoinflammation and autoimmunity, and more recently we are seeing more reports of patients with inborn errors of immunity who present with the spectrum of immune dysregulation that includes immunodeficiency, inflammation, and also atopy. So here shown is how canonical NF-κB pathway is heavily regulated by post-translational protein modifications, including ubiquitination and phosphorylation. Monogenic autoinflammatory diseases have been linked to mutations in many genes that constitute the canonical NF-κB pathway. So autoinflammatory diseases that result from dysregulation in the ubiquitin-proteasome pathway can result from a defect in ubiquitination, and here belong LUBAC deficiencies, this new disease that I will talk about later on, and RIP kinase-associated diseases, or they can result from a defect in deubiquitination, and here belong hyperinsufficiency of A20 or rotulin deficiency, or they can result from a defect in proteasome function. So let's start with diseases that result from a defect in ubiquitination. So LUBAC complex or linear UB assembly complex is E3 ubiquitin ligase, which has a role to attach MET1 or linear ubiquitin chains to different substrates such as RIP kinase 1, RIP kinase 2, MyD88, etc. And this step is essential for the stabilization of different receptor signaling complexes such as TNFR, IL-1 receptor signaling complex, etc. So LUBAC is composed of three subunits: the catalytic subunit HOIP and two scaffold protein HOIL1 and sharpin. So patients that have LUBAC deficiency, HOIP and HOIL deficiency, present with severe immunodeficiency. This phenotype is potentially lethal. However, in addition to severe immunodeficiency, they also present with autoinflammation, as shown here, with this strong gene expression inflammatory signature in their peripheral blood mononuclear cells. They also present with amylopectinosis, which basically results from the intracellular glycogen deposition, so they present with muscle weakness. And one patient, adult patient with a HOIP deficiency, also presented with severe lymphangiectasias. So LUBAC is essential for the regulation of NF-κB signaling. However, it has cell-specific functions as shown here in this experiment. So basically, stimulated patients' fibroblasts and B cells have a compromised NF-κB activity as shown here in this immunoblot, where you can see lower levels of phosphorylated IKKs or higher expression of the inhibitory protein IκB alpha in patient cells with HOIP or HOIL deficiency. In addition, their EBV transformed B cells are hyperresponsive to stimulation with TNF or IL-1 beta cytokine and produce less IL-6 cytokine. Interestingly, their peripheral blood mononuclear cells are hyperresponsive to IL-1 beta stimulation, and they produce more pro-inflammatory cytokines IL-6, which perhaps explain features of inflammation in this disease. Here shown is the patient with the sharpin deficiency who is a homozygous for a frameshift mutation in this gene. So similar to HOIP and HOIL deficiency, sharpin deficiency leads to destabilization of the LUBAC complex and impaired NF-κB signaling as shown here in this reporter assay. However, the associated phenotype is not as severe as is seen in patients with HOIP and HOIL deficiencies. Certainly, it's very different than what is observed in sharpin knockout mice who present with severe skin inflammation. Interestingly, this patient doesn't have clinically obvious immunodeficiency, rather presents with the features of inflammation, in particular with the joint inflammation and also GI inflammation. The inflammatory phenotype is more prominent in synovial fluid than in peripheral blood samples of this patient. Sharpin deficiency causes also hypersensitivity to TNF-induced cell death by apoptosis, as shown here in this vitro experiment, where you can see that cells, HOIP knockout or HOIP deficient and Sharpin deficient cells, upon treatment with TNF and cyclohexamides, are more prone to cell death, and this phenotype, cellular phenotype, could be blocked only with the treatment with the apoptosis inhibitor ZVAD, and was not blocked by the treatment with the necroptosis inhibitor. Currently, experiments are being done in patients' primary cells. Here is another disease that results from a defect in ubiquitination. This novel disease is named VEXAS and is caused by somatic mutations in the X-linked gene UBA1. UBA1 encodes the major E1 ubiquitin activating enzyme, which is required to initiate ubiquitination. So these patients present with a severe systemic inflammatory phenotype, as shown here in these images, with neutrophilic infiltrates in their lungs, with vasculitis, neutrophilic dermatosis, relapsing polychondritis. They have very active bone marrow, also present with bone marrow dysplasia, and they also have these highly specific vacuoles, which are found in their bone marrow precursor erythroid and myeloid cells. If you would like to hear more about this disease, please attend the presentation by Dr. David Beck tomorrow morning. VEXAS is caused by missense mutations that affect the methionine 41 amino acid residue, which initiates the translation of the canonical cytoplasmic isoform of UBA1. VEXAS is the late onset disease and is associated with a high mortality. So about 40% of patients in this cohort of 25 patients died from disease-related causes, either due to respiratory failure or progressive anemia or complications of treatment. The VEXAS-associated somatic mutations originate in multipotent hematopoietic stem cells, and they are found at the variable allele frequencies, typically in more than 50% in peripheral blood myeloid cells, but not in lymphocytes or fibroblasts. VEXAS mutations affect the translation starting at methionine 41 amino acid residue, and defect of this initiation of translation results in the loss of canonical isoform of UBA1, which is highly expressed in hematopoietic cells. In the absence of methionine 41, translation starts from the downstream methionine codon at the position 67 and results in the expression of a novel shorter isoform of this protein, which we name UBA1C. This isoform C is catalytic dead and cannot initiate ubiquitination process in mutated cells. Thus, in non-mutant cells, there are both nuclear and cytoplasmic form of UBA1, as shown here with these purple and orange dots, while in mutated myeloid cells, the cytoplasmic isoform is replaced with a non-functional isoform C, which is shown here with the green dots. Due to deficiency of functional cytoplasmic isoform of B of this enzyme, mutant myeloid cells have decreased level of polyubiquitinating proteins and increased level of free ubiquitin. Now, I would like to say a few words about recently described monogenic diseases that are related to dysregulation in the canonical NF-κB pathway. So here shown is the TNF receptor signaling complex 1, which is induced upon cell stimulation with TNF. RIP kinase is one of the central molecules in this pathway, and RIP kinase ubiquitination ultimately determines the difference between the cell survival signals and cell death signals in the cell. Ubiquitination of RIP kinase with K63 and linear ubiquitin chains promotes inflammatory and pro-survival signals in the cells. In cells where these pro-survival mechanisms are compromised, the balance will be shifted towards cell death pathway, either by formation of this pro-apoptotic RIP kinase 1, FAD caspase 8, caspase 10 complex, which is also known as complex 2, or by activation of necroptosis through this RIP kinase 1, RIP kinase 3, MLKL containing complex, which is known as necrosome. Pathogenic variants in RIP kinase can be inherited either as a recessive loss-of-function mutations, which are shown with the green color, or dominantly inherited mutations, which are shown in red color. Heterozygous missense mutations affect the cleavage of RIP kinase 1 by caspase 8, which is important for the attenuation of the RIP kinase activity. Thus, these mutations are essentially gain-of-function RIP kinase variants. RIP kinase patients' primary cells produce less IL-6 and TNF cytokines. However, they secrete more IL-1 cytokine. So defect in lymphocyte differentiation and decreased production of these pro-inflammatory cytokines likely explain the immunodeficiency in this disease. The increased production of IL-1 beta cytokine most likely explains the features of arthritis and intestinal inflammation in this disease. RIP kinase deficient cells are more sensitive to cell death by necroptosis as shown in these experiments. So TNF stimulated primary fibroblasts, and also it was shown in epithelial cells, and LPS stimulated RIP kinase deficient THP1 cells die more than control fibroblasts or cells that are transfected with the wild type RIP kinase construct, and this cellular phenotype could be only rescued upon the treatment of THP1 cells with inhibitors for necroptosis, and was not rescued upon the treatment of these cells with ZVAD, which is the apoptosis inhibitor. Essentially, this means that these cells are dying by necroptosis and not by apoptosis. RIP kinase variants that affect the protein cleavage by caspase 8 promote activation of RIP kinase and lead to autoinflammatory disease, which is characterized by intermittent fevers and lymphadenopathy. So these mutations are essentially gain-of-function mutations, and they can be inherited either as a de novo mutations in sporadic cases or can be propagated as a dominant trait in some families. These patients have a good response to treatment with IL-6 inhibitors. So peripheral blood mononuclear cells from CRIA patients have a strong inflammatory signature both for NF-κB and type 1 interferon-stimulated genes, as shown here in this RNA-seq data, and they release more pro-inflammatory cytokines. CRIA patients' myeloid cells are hypersensitive to cell death by apoptosis and necroptosis, while patients' primary fibroblasts are resistant to TNF-induced cell death, as shown here in this experiment. So what you can see here is that the cell viability is lower in TNF-stimulated PBMCs of patients, which are shown here with the pink bar, compared to control cells, and this cellular phenotype is rescued either by the treatment with apoptosis or necroptosis inhibitors or a combination of them. Similar observation was also made in knock-in mice with the RIP kinase CRIA-associated mutation. Mice homozygous for this mutation died during embryogenesis, and embryonic lethality can be prevented only by the combined loss of caspase 8 and RIP kinase 3. Mutant mouse embryonic fibroblasts, or MEFs, are sensitive to TNF-induced cell death, and genetic loss of RIP kinase 3 or RIP kinase 1 kinase activity significantly reduced TNF-induced cell death in these MEFs. Now I would like to say a few words about diseases that result from a defect in deubiquitination. So DABs are essential negative regulators of inflammation, and the removal of different polyubiquitin moieties leads to destabilization of various receptor signaling complexes. Here shown is the TNF receptor signaling complex, and eventually to the pathway attenuation. So patients that have haploinsufficiency of A20 or HAT20, they carry heterozygous loss-of-function mutations, typically nonsense or truncating mutations, which leads to defective K63 DAB activity of this protein, and also affects K48E3 ligase activity of A20, and impair also binding of A20 to UB chains. As a result of that, patients accumulate K63 ubiquitin chains in their stimulated fibroblasts, and we show the same thing in their stimulated peripheral blood mononuclear cells, and that leads to the constitutive activation of the NF-κB and NLRP3 pathways, as shown here. So again, here you can see higher levels of phospho-IκB alpha, lower expression of IκB alpha inhibitory protein, and higher levels of phospho-IkK proteins, which are the hallmark experiments or biomarkers for the constitutive activation of the NF-κB pathway. These patients respond well to treatment with cytokine inhibitors, and one patient also was cured by hematopoietic stem cell transplantation. mutation. Autolipemia is a very similar disease, except that it's recessively inherited, and these mutations, loss of function mutations, cause defect in linear DAB activity of this protein. What you see in patient-stimulated cells, again, is the excess of linear UB chains, and basically this leads to the constitutive activation of the NF-kappa B and MAP kinase pathways. These patients also respond well to treatment with various cytokine inhibitors, and one patient was treated with hematopoietic stem cell transplantation. Autolin deficiency in myeloid cells makes these cells hyper-responsive to TNF stimulation. They release more TNF cytokines and are sensitive to TNF-induced cell death. In contrast, autolin deficiency in primary dermal fibroblasts makes these cells hyper-responsive to TNF stimulation, which is a compensatory mechanism to downregulate inflammatory pathways in non-hematopoietic cells. Finally, I would like to say a few words about autoinflammatory diseases that result from the defect in the proteasome function. So PRAS patients, or proteasome-associated autoinflammatory syndrome, also known as CANDLE, these patients carry biallelic loss-of-function mutations in various subunits of the proteasome or immunoproteasome complex, which is essential for protein degradation and production of MHC class I restricted T-cell epitopes. While the constitutive proteasome is expressed in all cells, the immunoproteasome is expressed in hematopoietic cells, although it can be induced in other cell types. Here shown is the COR20S constitutive proteasome, which is functionally inactive until it associates with many regulatory proteins to make up functional 26S proteasome. The immunoproteasome differs from the constitutive proteasome in that the three of the constitutive proteasome subunits are replaced by different subunits, beta 1I, beta 5I, and beta 2I subunits. PRAS can also result from loss-of-function mutations in regulatory proteins. So these patients can have either heterozygous dominant negative mutations in the chaperone protein POMP, and you will hear more about this disease from Dr. Cecilia Poli, or they can have biallelic loss-of-function mutations in this regulatory protein PSMG2. Thus, because there are 7 subunits that make up for the proteasome and immunoproteasome, and there are many, many regulatory subunits that regulate the assembly of the functional proteasome, it is very likely that more pathogenic variants will be identified in other subunits or in other regulatory proteins in the coming years. Essentially, these patients have defect in proteasome-mediated protein degradation, which causes enhanced endoplasmic reticulum stress and also activates unfolded protein responses, and that ultimately leads to increased type 1 interferon signaling in mutated cells. This is probably not the only mechanism of the disease, but this is what has been known thus far. Because these patients have strong type 1 interferon signature, they respond well to treatment with JAK inhibitors. This is a very severe phenotype that present early in life. Many of these patients carry actually biallelic loss-of-function mutations in the PSMB8 gene, which encodes this beta-5I subunit, catalytic subunit of the immunoproteasome. Finally, I would like to say a few words about this disease, which I find very interesting, and that's a click syndrome. So while patients with the POMP deficiency in the major isoform of this POMP chaperone present with severe immunodeficiency and neutrophilic dermatosis, patients with the click syndrome are homozygous for one base pair deletion in the 5' UTR keratinocyte-specific isoform of this protein, and they present with a very different phenotype manifesting with this palmoplantar keratoderma, hyperkeratotic plaques, and so on. Interestingly, they have no features of systemic inflammation. So what is the exact mechanism of the disease in click syndrome? It's not clear. Supposedly, as a result of this proteasome insufficiency or deficiency in keratinocytes, these cells also upregulate ER and UPR responses. However, that hasn't been experimentally shown. So in conclusion, I would like to say that dysregulation in the ubiquitin-mediated signaling and protein degradation are emerging pathways in the pathogenesis of autoinflammatory diseases. The mutant proteins can lead to cell-specific cellular phenotypes, as I discussed on the examples of immune cells versus fibroblasts. Cell death pathways, either by apoptosis or necroptosis or both, contribute to the TNF-driven pathogenesis of many autoinflammatory diseases, and we should be looking into somatic mutations, or should be considering somatic mutations as a cause of disease in patients with various immunological disorders. Finally, I would like just to make some acknowledgement to many people from the NIH intramural program, including Human Genome Research Institute, our postdocs that are doing basic science work, our clinical team, our mentor, Dr. Daniel Kastner, to many collaborators from other NIH Institute, NIMS, NIID, Department of Laboratory Medicine, to our international collaborators for working with us on different diseases and sending their patients, and also to patients and their families. And finally, I would like to thank you all for your attention.
Good afternoon, everybody. I'm very pleased this afternoon to give you a presentation on an adaptation program that we think is involved in controlling the response to defect in the proteasome. My laboratory has been interested for several years already in understanding how malfunctioning cells that have defect within their function can restore their homeostasis through adaptation program, and how these adaptation programs are linked to inflammation. Over the years, we have used as a model to study stress response the study of the HIV protease inhibitors. These drugs were designed to target the HIV protease, but they also have off-target effects in humans, including some atypical anti-cancer properties. So the mechanism of action and the targets in mammalian cells were not known. So we developed a program that was aimed at identifying the pathway that are regulated by treatment of the HIV with the HIV inhibitors, including nalfinavir, as well as cellular target of these drugs. And I'm not going too much in details, but just to mention, we identified several pathways that were related to translation inhibition that were affected by nalfinavir. And this is common for molecules and conditions that affect the homeostasis of cells, because you need to slow down translation to repair and restore homeostasis. We also found impact on activation of aspects of the innate immune system, such as the M2-inflammasome. And just to make a long story short, among the targets of nalfinavir we identified, there was this protease ESID-2020 that is involved in the maturation of the lamine, and it maintains the nuclear envelope integrity. And once this nuclear envelope is destabilized, you have transient exposure of nuclear DNA that can activate inflammatory pathways such as the M2-inflammasome. But we were very excited by these findings, but when we looked at the role of ZAMS24 in the activation and the regulation of translation machinery, we couldn't link those two. So this told us that there is another target of nalfinavir, and we developed several approaches to identify those other targets. And today I'm going to focus on a bioinformatic approach where we looked at proteases in humans, in mammals, that were very close to the HIV protease. And we identified a family of proteases that were very interesting to us, in particular, this protein here called DDI2. And we focus on DDI2 because this protein is ubiquitous, and phenotype we had been investigating with the HIV protease inhibitor was also ubiquitous. So you find it almost in every cell type we looked at. But DDI2, we don't know much on the function, so our working model is that DDI2 is activating some kind of stress response pathway that leads to shut down of the translation machinery or decrease of translation efficiency within the cells. So what do we know about DDI2? At the time when we started this project, there was not much known. We knew it was an osmotic protease. We generated a knockout mouse line, and it turned out that knockout mouse was embryonic lethal, suggesting that this protease had some kind of important function during development. And we had some in vitro evidences that there was some kind of interaction, functional interaction with nalfinavir. So in order to better address the function of this protease, or to identify the function of this protease, we generated a conditional knockout line. And those are these DDA2 flux line that we crossed with the albumin-free mice, and this led to the deletion of DDA2 in the liver of mice. When we looked at, we phenotyped those animals, there was no much phenotype at first. So we sequenced the livers and looked at the gene expression within liver in order to identify genes that were differentially affected by DDA2 deficiency. And the top hits were these MT1 and MT2 genes. Those are detoxifying enzymes that are, for example, involved in the response to cadmium in the liver, and they are very important in regulating detoxification reactions. So when we looked at the literature and we looked at the genes that were affected by DDA2, we realized that there was a significant overlap with published reports on the NRF1 transcription factors. For example, in the NRF1 knockout animals in the liver, there is the same phenotype in terms of MT1 and MT2. So it is kind of suggested to us that maybe there was a link between the DDA2 pathway and the NRF1 pathway. So what is NRF1? NRF1 is a transcription factor. It's a member of the NRF2 and NRF3 family. So the NRF2 transcription factor is very well studied. It's a transcription factor involved in oxidative stress response, and it has a connection to inflammation, cancer, etc. So it's been heavily investigated. But mechanisms of activation of NRF1 are different than the NRF2, and they are poorly understood. Interestingly, the knockout mice, the knockout NRF1 animals, they are mononucleatal, similar to the DDA2 knockout animals. And when people looked at specific function of NRF1, there were several studies that suggested that NRF1 is a regulator of proteasome subunit gene expression. So it seems to be involved, it seems to be able to trigger the transcription of the proteasome subunits and therefore promote the production of new proteasome. It's also a target of ERAD, and it's actually located within the endoplasmic reticulum, and then it's retroslocated in the cytosol by the ERAD machinery, and it's normally targeted for degradation by the proteasome. So we, looking at the literature, we build a model where NRF1 within the ER is retroslocated in the cytosol and where it's generally degraded by the proteasome. But if you have proteasome defects or proteasome inhibition, the NRF1 is stabilized in the cytosol, and there it can get cleaved by a protease. And this was shown before. There were like studies showing that NRF1, in order to get active, needs to be cleaved by a protease, but the protease identity was unknown. And because we had this link with DDA2, it was easy to suggest that DDA2 was the protease. And I'm going to show that actually in a minute. And once DDA2 cleaves NRF1, or the putative protease cleaves NRF1, NRF1 triggers the expression of proteasome subunits that then restore proteasome function. That was the working hypothesis and the working model. And I'm going to show you some data that confirm these working hypotheses. First, we deleted DDA2 in several cell types or in the mice and knockout mice we have. And in each conditions, we could observe that if you treat those cells or those mice with bortezomib, you have cleavage of NRF1. If you don't have DDA2, you don't have the cleavage of NRF1. And this is consistent in different clones and different cell types. Other groups have had similar findings. There was a couple of papers, including recently, that at least in vitro and in C. elegans suggested similar phenotypes. When we expressed a version of DDA2 that is mutated within the active domain, within the protease domain, we observed that we lose the ability of cleavage of NRF1. And this is caused by the fact that DDA2 is a dimer, it forms a nomodimer, and therefore if you have mutated DDA2, it can dimerize with wild-type DDA2 within the cells, and then it blocks the function of the wild-type DDA2. So it shows you that the protease activity is required for DDA2 function, and the protease activity is required for the cleavage of NRF1. We did a proteomic approach to identify the cleavage site of NRF1 by DDA2. And then we mutated that site, and this is the experiments that you test those mutation. So this is in a control cells. NRF1 is cleaved. If you have DDA2 deficiency, NRF1 is not cleaved. And if you transfect the mutated version of NRF1, the cleavage is reduced. If you compare these to these, you see that there is a clear decrease in the cleavage. This is a longer exposure of this immunoblood, so that's the cleavage in the wild type, and that's the cleavage in the mutant NRF1. So the cleavage is very, very reduced, but we always have a residual cleavage that we don't explain for now. But we don't think it's another cleavage site. We just think that there is a residual activity within this region. We tested the nelfinavir as an inhibitor of this cleavage, and we could see some inhibition. This is one of the experiments that showed that basal cleavage of NRF1, and we can see that nelfinavir is the most potent HIV protease inhibitors, but we could observe some activity with lopinavir, saquinavir, and ritonavir. I have to say that the concentration of nelfinavir that are functional here are pretty elevated, and we don't think it's very strong inhibitors actually of DDA2. We need to work on that a little bit more to make sure that this is physiologically relevant. So most of the studies then I'm going to focus on that work that focus on DDA2 are studying DDA2 deficiency rather than an nelfinavir impact on DDA2. So we needed a system to kind of demonstrate that this pathway is relevant. So we decided to study the DDA2 and NRF1 pathway in the context of multiple myeloma. Multiple myeloma is a tumor type of that uses inhibition of bortezomib as a first line of treatment, and with some good effect. So we decided to use that model and see whether the DDA2 pathway could be involved in that response. So this is again a summary of our working model. So NRF1 is present in the endoplasmic reticulum, where it's glycosylated. It is retranslocated in the cytosol by the ERAD machinery, and then it's deglycosylated in the process, ubiquitinated and degraded by the proteasome. If you have a defect in the proteasome, NRF1 accumulates in the cytosol of cells, and therefore it's accessible for cleavage by the DDA2 protease. So DDA2 will proteolytically cleave NRF1, and this cleavage will release an active form of NRF1 that it will enter the nucleus, and where it can actually switch on the transcription of proteasome subunits, and this allows the increase in the proteasome amounts and restores some proteasome function. So we deleted DDA2 in multiple myeloma, and what we can see is that once you delete DDA2, you don't see the cleavage of NRF1 anymore. So this is bortezomib treatment in control cells. NRF1 is cleaved, DDA2 is present. If you delete DDA2, as you can see here in these clones, you don't have DDA2 anymore, and you don't see cleavage of NRF1 anymore. What you can see is that NRF1 is not present once you delete NRF1. We use these NRF1 knockout cells as a control to compare the impact of DDA2 deficiency to NRF1 deficiency. When we look at proteasome induction, what you can see is that if you focus on the black bar, if you treat cells with bortezomib, you have most of the proteasome subunits increase in expression. This is part of this proteasome response pathway that we are trying to define. And if you have deficiency or deletion of DDA2 or NRF1, you don't see anymore such a striking induction of the proteasome subunits. And this is kind of confirmed that in multiple myeloma at least, the DDA2 and NRF1 can respond to bortezomib, and this response is dependent on both DDA2 and NRF1. Does that translate in an impact on the toxicity mediated by bortezomib? So multiple myeloma cells are sensitive to bortezomib treatment. And what we did here is that we treated these cell lines, either control cell line in black or DDA2 and NRF1 knockout cell lines with increasing doses of bortezomib. And what you can observe is that deficiency in DDA2 and NRF1 decrease the viability of the cells and therefore increase susceptibility to proteasome inhibition. This is quantification of those results that showed from multiple experiments that is a very significant impact of DDA2 deficiency when it comes to multiple myeloma susceptibility to bortezomib treatment. And then in order to confirm that this is really mediated by DDA2, we reintroduced in DDA2 deficient cells DDA2 using a CRISPR-resistant DDA2 under the control of doxycycline promoter. So what you can see here, these are again the control cell lines. NRF1 is cleaved upon treatment with bortezomib. If you have DDA2 deficiency, NRF1 is not cleaved, but it accumulates as a proform. And then if you have the same cell line here, but reconstituted with DDA2 under the control of doxycycline, if you don't have doxycycline, you have no phenotype, so there is no DDA2 and no cleavage. If you had like doxycycline, it reintroduces DDA2 in the cell, and it reintroduces the cleavage of NRF1. And then we tested here the response in terms of viability and response to increasing doses of bortezomib, of treatment with bortezomib. And what you can see here, in black, again, you have the control cells. In red, you have the, in orange, sorry, you have the DDA2 knockout cells here that are more susceptible to treatment with bortezomib. And then in red, you have the control cells reconstituted with the DDA2, but not treated with dox, so DDA2 is not expressed, and therefore these cells behave exactly like the knockout cells as expected. But as soon as you reintroduce DDA2 with the treatment with dox, you have a response that is similar to the control cell line. So you have less susceptibility to bortezomib treatment. This again confirms that the response to proteasome inhibition is mediated here in terms of viability, is mediated by the DDA2 and the NRF1 pathway. So basically what those data suggest, again, is this is the confirmation of the initial model we had, is that as soon as you have a defect in the bortezomides, for example, as soon as you have a defect in the proteasome, for example, upon treatment with bortezomib, NRF1 accumulates, it's cleaved by DDA2, and that leads to the upregulation of proteasome subunits and restoration of proteasome function. So all the studies were done using chemical inhibitor, biochemical pharmaceutical inhibitor of the proteasome. And so we would like to now move and ask whether this could be involved in diseases that are linked to defect in the proteasome. And this is we're trying to focus on, and this is work in progress. We don't have data now, but I'm just showing you our main hypothesis. When you have mutation in the proteasome subunits or the immunoproteasome, a proteasome that is expressed constitutively in hematopoietic cells, or mutation within the genes involved in the assembly of the proteasome, you get decreased proteasome activity within those patients, and those translate with an autoinflammatory phenotype that is mostly relying on a type 1 interferon response deregulation. So you have increased type 1 interferon response in those patients. Mechanisms are not well understood. What we are going to ask, because this provides us with a model for studying proteasome defects, we would like to interrogate whether this pathway is increased in those patients and is activated by the decreased proteasome function, and whether it plays a role maybe in decreasing symptoms or decreasing the phenotype in some patients. We would like also to test, because this disease here, this proteasome-associated autoinflammatory syndrome is associated with the upregulation of type 1 interferon. We like to address, and other cytokines. We like to address whether NRF1 as a transcription factor, if it's really activating this disease as we expect, whether NRF1 could contribute to this pattern and to this pro-inflammatory response, because there are some evidences that some of the target genes of NRF1 could actually be pro-inflammatory. So I would like to leave you with this model, this idea, this model here. I hope we'll be able soon to provide you with more information on this disease. I would like to thank the people involved in this study in my laboratory. Several groups, several people have been working over the years on this DDA2 project. Sergio is working mostly in the mice. Melanie is working mostly on the multiple myeloma project, and Claire is working on the biochemistry of NRF1 DDA2. I would like to thank you all for your attention.
So first of all, I would like to thank the organizers for inviting me to this wonderful meeting among such amazing speakers. I have nothing to disclose. So interferonopathies are very heterogeneous. They have very different clinical presentations, even though they all cause an upregulation of interferon genes. But some of them, such as SAVI and COPA, with the pulmonary compromise, do have some things in common that perhaps point to a common immunopathology that is perhaps also tissue-specific. So interferonopathies can be caused by defects in distinct pathways, such as the RNA-DNA sensing pathways, causing sting and iCardiac tears, proteasome dysfunction causing candle, and others such as COPA and ADA2 deficiency. So the underlying mechanisms for most of the interferonopathies remained unsolved, and there's many questions about how are interferons produced. So are they directly pathogenic? If not, what drives autoinflammation and specific tissue damage? If yes, do interferons drive all clinical manifestations? Also, if proteasomes, sting, COPA, and iCardiac tear genes are ubiquitously expressed, why is damage tissue specific? So addressing all these questions is important for therapies. So although these are all mainly interferonopathies, we know that interference may also drive the upregulation of other autoinflammatory pathways, or perhaps the same defect that is driving the interferonopathy can also drive dysregulation of NF-kappa B pathways or other autoinflammatory pathways. In fact, Adriana de Jesus described very nicely in this paper that NF-kappa B is dysregulated in a subset of interferonopathies such as NEMO, MDAS, LRBA, and others. And this is important because identifying this immune dysregulation can help decide on therapy. We know that interferons are pathogenic up to some extent because therapeutic inhibition of interferon ameliorates the clinical phenotype. So this supports pathogenicity. They're beneficial in half of the patients with Kendall. They show partial improvement and prevent progression in patients with SAVI. And patients with iCardiac tear also present partial improvement with baricitinib. There's few reports suggesting benefit in COPA patients and other more recently described interferonopathies. So understanding what underlies and accompanies the interferon signature in each specific interferonopathy is paramount for therapeutic approaches. So for example, COPA syndrome, that is an autosomal dominant autoimmune disease associated to ER stress and characterized by pulmonary hemorrhages and inflammatory compromise of the joints, is caused by mutations in COP-A that is in charge of bringing vesicles from the Golgi to the ER. So there's a defect in the vesicular transport from Golgi to the ER, and this causes ER stress, which is depicted here by increased BIP in pulmonary biopsies. They are associated to an increased interferon signature. And one could think perhaps your stress is leading to this interferon signature, but what is the precise mechanism? So Alex Lepley in this paper beautifully showed that STING, because there's a defect in GOLD2ER transport, is retained in the ERGIC, and this is leading to a permanent activation of STING. So STING here is mediating the increased interferon in COPA and perhaps mediating the immunopathology. And this could explain why some COPA patients have some features such as pulmonary compromise that is similar to patients with STING. So what happens with proteasome dysfunction and interferonopathies? So there's different types of proteasomes. Proteasomes are made up of 2 alpha rings and 2 beta rings, and these are assembled thanks to proteasome assembly chaperones such as POMP in PAC1, PAC2, and PAC3 and 4. Proteasomes are assembled, and standard proteasomes have standard beta-catalytic subunits, and in immune-stimulated proteasomes exchange these beta-catalytic subunits for specific immunoproteasome catalytic subunits. Then these are coupled with regulators to form the hybrid 26S proteasomes. So mutations within the proteasome subunits cause PRAS or CANDLE, chronic autoinflammation with neutrophilic dermatosis, lipodystrophy, and elevated temperature. There are 2 genetic mechanisms of disease for PRAS. One is autosomal recessive, and it can be digenic, meaning that there's 2 different mutations in 2 different proteasome subunits leading to CANDLE. And the other one is autosomal dominant, loss of function mutations in POMP, which is the proteasome chaperone, and which is believed to be dominant negative. So mutations in all of these proteasome subunits can lead to CANDLE, as well as mutations also in RPT5, that's one of the alpha subunits, and mutations in the proteasome assembly chaperones, such as POMP and PSMG2, and they all lead to similar phenotypes. More recently described mutations in RPN5 encoded by PSMD12 lead to a neurodevelopmental disorder with autism without inflammatory manifestations. In mutations in PSMC3 encoding for RPN5 cause deafness in congenital cataracts with developmental delay without evident autoinflammatory features. They do have calcifications of the elbows and the knees, but they don't have any clear signs of autoinflammation. So this led to the hypothesis that there can be distinct phenotypes depending on what proteasome subunits are affected. So mutations in the 20S core particle will be causing CANDLE, and mutations in the 19S regulatory particle seem to be causing neurodegenerative and neurodevelopmental disorders. However, more recently described autosomal recessive mutations in PSMB1 encoding for the constitutive proteasome catalytic subunit beta 6 cause microcephaly, intellectual disability, developmental delay, and deafness with short stature without autoinflammation. So this suddenly challenged our hypotheses about the catalytic subunit and the regulator, and more research needs to be conducted in patients with mutations in this different subunit to understand really what is driving disease in different tissues. So we were interested in the question of what precisely drives interference in proteasome deficiency PRAS. So we know that proteasome deficiency leads to the accumulation of ubiquitinated proteins, ER stress, activation of the UPR response, and upregulation of interferon type 1 genes. So we used, to study these mechanisms, we use autosomal POMP deficiency characterized by autoinflammation, neutrophilic dermatosis, high autoantibody titers, and immunodeficiency leading to mainly sepsis and recurrent respiratory infections, including opportunistic infections by PJP and mycobacteria. So as I just mentioned, POMP is a chaperone for proteasome assembly. To investigate these different mechanisms, we used different inhibitors of the UPR sensors and STING to identify which was the driver of interferonopathy. So we use one events to inhibit PERK, 4-MUHC to inhibit IRO1, and H151 to inhibit STING. And this is the work of my student Valentina Harur while she was working with our collaborators Alke Kruger and Frederick Epstein at Greifswald. So what she did is she tried these different inhibitors in patient PBMCs, and neither STING, IR1, or PERK inhibitors modified the interference signature of patients' PBMCs. At that time, Seth Masters and Sophia Davidson were working on a hypothesis that misfolded IL-24 could be accumulated due to proteasome dysfunction in the cytoplasm, and this could stimulate PKR, which is a known stimulator of EIF2 alpha phosphorylation, which leads to the homeostasis of ER stress. And we decided to test this hypothesis in our patient cells. So we used C16, which is a PKR inhibitor, and what we observed is that the PKR inhibitor ameliorated the interference signature, bringing it back to normal. And we observed this in both of our patient cells. And what you can see here is how C16 PKR inhibitor dramatically dropped the interference score in both patient cells, indicating that the interference signature in POMP deficiency and perhaps in other patients with PRAS is mediated by PKR. So we know that proteasome dysfunction leads to UPR activation, and UPR activation is known to activate NF-kappa B. We knew that the UPR was increased in POMP deficient cells. They have increased ATF6, XBP1 spliced version, and phosphorylation of EIF2 alpha in the PERK pathway. But they had NF-kappa B dependent cytokine release, such as TNF alpha, was basically absent in both patients. So we hypothesized that because POMP it participates in the assembly of not only the standard proteasomes, but also the immunoproteasomes, perhaps these defects could lead to an impaired capacity of the proteasomes to degrade I-kappa B alpha and trigger NF-kappa B signaling. And this could perhaps explain immunodeficiency in our patients. However, when we looked at I-kappa B alpha degradation in patient cells, we saw that I-kappa B alpha degradation was enhanced. And when this was significant for both patients. So NF-kappa B was, in fact, as in other proteasome diseases, hyperstimulated. And this could also be leading to some of the features that we saw in the patients. When we transfected cells with an NF-kappa B luciferase construct and mutant and wild-type POMP, we observed the same. The mutant POMP led to an increased NF-kappa B signaling. This also supports the dominant negative mechanism by which POMP mutations are leading to PROS. So when we used an interferon inhibitor and looked at NF-kappa B activation, we saw no difference. However, when we used C16 PKR inhibitor in this system, we observed that NF-kappa B, as well as the interference signature, signaling was decreased was decreased. So PKR not only was involved in the activation of the interferon signature, but also in the activation of NF-kappa B. So in conclusion, mutations in the catalytic versus regulator proteasome subunits lead to distinct clinical phenotypes, and tissue specificity or redundancy of some subunits could explain this phenomenon, and this needs to be further studied. It is unclear if the brain-specific inflammation is involved in proteasome-associated neurological disorders. Consistent with what has been reported for other proteasome deficiency, both NF-kappa B and interferons are dysregulated in POMP deficiency. Exacerbated interferon and NF-kappa B pathways seem to be driven by PKR. But the underlying mechanisms of immunodeficiency and POMP deficiency still remain to be elucidated. Because our patients were so immune deficient, and we did not want to use JAK inhibitors and decided to transplant them. So what I would like to show you is the results of the transplant more than 2 years after these patients have been transplanted. So they were both received stem cell transplant after reduced intensity conditioning at 40 and 30 months, respectively. Both received 10 out of 10 bone marrow grafts. Both patients engrafted before day 21 with 100% donor chimerism in all cell lines and only had mild skin GVHD. As you can see here, the proportion of T cells that was markedly decreased before transplant normalized after transplant in both patients. Here. And the cytokine release defect that we observed in both patients also normalized post-transplant for interferon gamma, IL-2, and the NF-kappa B dependent TNF-alpha. When we looked at the accumulation of ubiquitinated proteins, we observed that these was comparable to normal individuals after transplant. So protein homeostasis was restored post-transplant, and this correlated with a significant decrease in an interferon signature comparable to normal individuals. So both patients are 4 and 2 years after transplant. They're alive and well with no signs of chronic inflammation. They don't have autoantibodies. They haven't had severe infections, and they even had flu without complications. And during this time, the patients have not developed lipodystrophy or dyslipidemia. And this is one of the patients as he started school last year before coronavirus hit us. So in conclusion, stem cell transplant with reduced intensity conditioning can effectively correct immunodeficiency and autoinflammation in POMP deficient patients. And perhaps this is applicable to other proteasome defects. Patient recovery with transplant, despite the ubiquitous expression of POMP, suggests that the phenotype here is mostly driven by POMP deficiency in immune cells. We do not know what will happen in the long term with the neurologic development of these patients, since POMP is also expressed in the brain. CD8 T cell recovery suggests a role for DCs that repopulate the thymus in promoting CD8 T cell differentiation, because although he has the same thymus with the same thymoproteasomes in cortical thymic epithelial cells, CD8 T cells normalized after transplant. Some final remarks. So interferonopathies are heterogeneous, and understanding the underlying mechanisms driving interferon response may improve therapeutic strategies. Additional immune pathways leading to autoinflammation may play a significant role in driving inflammation and interferonopathies, and they could be interdependently targeted, so they need to be identified in these interferonopathies. Inborn errors of the proteasome function lead to distinct phenotypes, and understanding the biology of the regulatory and catalytic subunits in different tissues will contribute to understand these differences and design therapeutic approaches accordingly. Finally, the identification of PKR as a driver of interferonopathies in PRAS opens new therapeutic possibilities. And with that, I would like to thank my mentor, Dr. Jordan Orange at Columbia University, Elke Kruger and Frederick Epstein at Greifswald, Dr. Caridad Martinez, that was in charge of the transplant of both of these patients. Lisa Forbes and Sarah Nicholas took care of the patients clinically. Seth Masters and Sophia Davidson for all the work in PKR. And Valentina and everyone in my lab for all their work and their constant encouragement. Also some funding sources. Thanks.
Thank you very much for all the speakers that just gave us a 3 wonderful talk regarding this pathway of protein. I'm going to go through some questions that we already received regarding, first of all, Ivona told, so we have a question for you regarding sharpening, whether has some cell-specific role been described, and can this explain why it is different, the phenotype between sharpening oil and Hoy patients?
Thank you for this question. So yes, this patient is indeed very interesting because we are really seeing not so strong inflammatory signature in peripheral blood, or not so much systemic inflammation. Everything is really happening at the tissue level. As you could see in my slides, primarily in GI tract and also in synovial tissues. So what we are doing currently is studying the effect of the sharpening deficiency in peripheral blood mononuclear cells and fibroblasts. These are the only cells that we have access to. It would be nice if we could have, access to other cell types, but that's what we can do at this time. And what was your second question? And why the phenotype is so different, or something like that, right?
Yeah, exactly. So
yeah, that's really peculiar, because this patient doesn't have obvious immunodeficiency. It doesn't look like it doesn't have phenotype which is observed in sharpening deficient mice. So we don't really have a good explanation for that. I mean, if it wouldn't be for this, the fact that this patient is a homozygous for the nonsense mutation, I would even worry whether this is really a true disease-causing change. But this mutation is nonsense, and basically creates the truncated protein. So it's just, peculiar why this patient has so different phenotype than other patients with Lubach deficiencies or a sharpening deficient mice. I don't have good explanation for that, but it's quite complicated.
Okay, we have a second question for you regarding the VEXAS patient. Yeah. And somatic mosaicism. Do you think that the age of onset correlate with the level of somatic mosaicism? And if so, is it possible that we might find patients that are younger, so with an onset in a younger age?
It is possible that we will find patients at the younger age. What will be the level of somatic mosaic mutations in these patients? We don't know. Probably it's going to be low. We are doing this type of analysis to see whether there is a progression, or whether you see basically more of these somatic mutations as the patients age. And there is evidence that, there is definitely higher allele frequency of these mutations in older patients. But we are also limited this is a relatively new project, and we are limited, with patient samples, prior to discovery of these mutations, so that we can really look at the progression of these mutations over the time. And the second question is?
I think this was it. So, the second question was regarding lung phenotype. So, if you can expand about the respiratory failure and the cause of that in the patient.
Okay, the only thing I know about the lung phenotype thus far is that basically it's caused by neutrophilic infiltrates in the lung, and I can't tell you anything else. So as a result of this mutation, basically primary patient myeloid cells are hyperactive, and they just infiltrate different tissues, including lung. They also present with a severe skin inflammation, but that's all that we know at this time.
Okay, thank you. So we have some questions for Cecilia Poli. The first question is if you see any therapeutic implication from Marco Cartorno for other drugs than anti-interferon or JAK inhibitor in general for the patient you described.
Yes, so because NF-kappa-B is also hyperactive, I wonder, and this is something that was not used in our patients, if they would respond to anti-TNFs, or some part of the phenotype could respond to anti-TNF inhibition. As far as I also have not used it in any patient with other forms of candle. So I cannot answer that question clinically, but if some of the phenotype is driven by NF-kappa-B hyperactivation, perhaps that could help.
Okay, and we have another question from Betsy Cleve. If the hematopoietic stem cell transplantation was challenging because of high interferon?
Yes, that's a very good question. These were patients that were hard to transplant, both in the conditioning phase and in the post-transplant period. They both had a very severe engraftment syndrome, but it was managed with ICU care and immunosuppressants, and they overcame it in a period of a week. And other than that, they did not have a severe GVHD other than mild skin GVHD. That's it. And they're both healthy. One of the boys is practically normal, like you couldn't tell he was ever sick. The other one, he was more severely ill, and very early on he had some cerebral infarcts during the neonatal period, so he has a developmental delay, and he's also short. He received a lot of steroids. So
I have a question for you, a similar question. Do you think there is space for other type 1 interferonopathies for hematopoietic stem cell transplantation?
I think there is. I think what is so interesting about these two patients is that they are well. So this tells us that most of their phenotype is driven by immune cells and not by the deficiency in other cell types. It remains to be elucidated what will happen in 10 years from now if they will have protein buildup in the brain and perhaps have, early onset dementia or something like that. I mean, these are things that we've hypothesized could happen, but they do have a very mild interference signature still. One of the patients does, but it doesn't seem to be clinically relevant. Now they are not immune deficient, one could use JAK inhibitors.
That is very interesting. So it links me to another question from Yannick Crow. He asked for a PSMB1 patient that you said they don't have a clear inflammation or inflammatory phenotype, but as you said, probably until you test them, you will not know whether if they have an interferon signature or not, for example.
Yeah, so I unfortunately I don't have any patients with PSMB1 mutations. Those so it was a collaboration, and it's not reported that they tested on the interferon genes. But I wonder, there's other Aicardi-Gouttiere patients, as Yannick mentioned, that do not have a peripheral interferon signature, yet they are an interferonopathy. So one wonders what's happening with the interferon gene expression in the brain tissue, and if this is playing a pathogenic role specifically in that tissue.
I don't know if you, Yannick, want to comment on that.
Yeah, actually, it was a point the other way around, really, that, I just don't think you can. I mean, whether interferon is driving the disease is another question, but you cannot, most AGS patients, classical AGS patients, they'd never go to a rheumatologist. Yeah. The rheumatologists have taken over this field now, but originally it was about neurologists. And these patients don't have fevers, they don't have ISRs, they don't have CRPs, they don't have low platelets. They're just a sort of static neurodevelopmental disease. And I think, so what you said was that the absence of an autoinflammatory phenotype challenged your theory about the proteasomal subunits, and I would say, well, I'm not sure you need to worry about the challenge because you don't, until you look, I just don't think as I say, I do worry about putting all these things together and that the interferon is driving everything. And Ivona actually made a couple of good points about some of these other disorders with the RIP kinase, I think, Ivona, where you showed that, or people have shown that there's various transcriptomic programs induced. But yeah, that was my point.
Okay, we have again a question from Yannick to Fabio Martinon regarding NRF1 function and NGLY1. So there is a disease that's caused by mutation in NGLY1, and this has been described to be mediated at least by NRF1. And do you think this can fit in your schema or in your data or not?
So thank you, Yannick, for the question. I think it fits in the scheme, but I have no explanation of the differences that probably occur here. So NGLY1 is required for NRF1 activation. If you don't have NGLY1, you cannot deglycosylate the transcription factor, and that transcription factor is not functional. So there is clearly a link between NGLY1 and NRF1. But when we have a defect in the proteasome, we have the opposite outcome. We have increased NRF1 activation. So it doesn't really explain the differences in the mechanism. So in that study, they showed that NRF1 contributes also to the maintenance of the mitochondria. And so you have a mitochondria defect. And that's what causes a type 1 interferon response in those patients. And we, at least for now, we haven't observed similar things in the context of proteasome defect. But NGLY1 is also involved in the ERAD pathway, and so it may lead to the accumulation of more proteins and may lead to the increased ER stress. So if there is an ER stress component in the context of proteasome mediated autoinflammation, it's possible that there is a link between the two there. And it's also possible that NRF1 defect impairs the proteasome in these patients because you don't have this adaptation program to restore proteasome function if you don't have NRF1. That could also be an explanation for some aspects of the inflammation observed there. But mechanistically, we have no evidence of how things work in NGLY1 deficiency for now.
Lovely, thank you.
Okay, another question for you, Fabio, is that could we expect a disease due to NRF1 mutation? So a phenotype caused by mutation in this gene.
So the NRF1, like the DDI2, so when we made the DDI2 knockout mouse, we were very surprised because it's embryonic lethal, and NRF1 is also very important during development. So complete deficiency in NRF1 is unlikely, but there may be some hypomorphic mutation or defect in the pathway. But for the moment, to my knowledge, nothing has been linked to DDI2 or NRF1 in humans. But yes, but there is also something that I didn't stress. This pathway is really an adaptation program pathway. It's not required for the normal amount or the basal amount of proteasome expression or function. But it's very important when you have a defect within the proteasome, then it allows to increase the amount of proteasome, and that's it's really an adaptation program, like showing that the proteasome is something plastic that can adapt to needs for more proteasome. So I don't know what will happen if you have a defect in the pathway. Yeah.
Then we have another question for Cecilia. If you can elaborate on the T cell function in your patient, because those patients suffer from mycobacteria, and also how the B cells were affected, because you show that they have increased immunoglobulins.
Yeah, thanks for those questions. They're both very important. So on the T cells, they didn't produce any interferon gamma in vitro upon stimulation. So I think this could have something to do with one of them getting a mycobacterial infection. They were also skewed. They were basically all CD4 naive T cells. They had very low percentage of memory T cells and very low percentage proportion of CD8 T cells, as one would sort of expect, because proteasomes are important for the thymoproteasomes as well, which participate in the CD8 CD4 differentiation. So one would expect if MHC class 1 is down, then you would have less CD8 T cells, and this is what we saw in these patients, and this could also account for the patient's immunodeficiency. With regards to the B cells, they were abnormal. The proportion of plasmablasts was greatly increased, and we even observed plasmablasts in the bone marrow. And I think this explains why these patients had very important autoimmunity. Even when they were a month of life, they had positive antiphospholipid autoantibodies and anti-beta-2 glycoprotein 1 autoantibodies, anti-thyroid autoantibodies present from very early on. So there was a B-cell dysregulation as well.
Thank you. So there is a question for everybody, I guess. So how are interferonopathies and COVID-19, dealing with COVID-19? I don't know if any of you had any experience of patient with type 1 interferonopathies infected by COVID-19. So none of our patients, fortunately so far, got infected. Our feeling is since the first weeks of the pandemic, we were very scared, because one of our first patients with SAVI, when we started treating her with JAK inhibitor, she actually suffered from 3 to 4 viral infections. It was influenza, then varicella, and then right after varicella, she got a coronavirus, a regular coronavirus. It was 3 years ago. And she went to very severe respiratory insufficiency and got intubated and then needed ECMO. So we are actually very scared about this, but so far I don't know about any report. I know there are actually clinical trial of JAK inhibitor in the treatment of COVID-19, but I'm not aware of the result of those. And we are personally kind of reluctant of the result, or very curious, let's say, of the result. But we don't know. This is our answer, at least, in our experience.
I can just add that we don't really have that many patients with type 1 interferonopathies, but based on what was published recently in the Science Journal, that type 1 interferon pathway is crucial for defense against coronavirus infection, you would think that these patients overall would do better, bearing in mind that their treatment, whether it's strong or milder, etc., can affect, their response to this infection. But theoretically, they should do better, right?
Mm-hmm. So we have 2 STAT1 gain-of-function patients that are not currently on a JAK inhibitor, and they both encountered COVID in their family members, and they didn't get sick.
Yeah.
So that's yeah.
Okay, that's reassuring, let's say. Thank you. And I have a question that is, I don't know if you can answer, but can you speculate on that? Is about COPA and incomplete penetrance. It's kind of, I think it's unique in the type 1 interferonopathies, or as far as I know, there are several families where you have patients with the mutation that are not at all affected by the disease. And can you speculate on that, Yannick or Cecilia or Ivona or Fabio?
I'm dealing with COPA mutations, or mutations that I find in this gene. I just don't know, whether they're pathogenic or not. So that's my experience with this gene.
But it's quite easy, Ivona, because there's a little hotspot. So it's very interesting. I think it is, you're right, Stefano, it is very interesting. And actually, I think if you kind of do, so of course, asymptomatic persons don't necessarily get tested in a family, but if you kind of look at the family trees that have been published and from the cohort, a small cohort of 6 or 7 families, I think we've got this something like a 20 to 30% non-penetrance rate for mutation carriers. Now, so what I think what's quite interesting is also in MDA5, IFEH1, we've seen high non-penetrance. So we've seen about 10-15% non-penetrance there. But there is a bit of a difference, which I find intriguing. I can't quite work out in what it means. I'd say the vast majority of people who are clinically non-penetrant with an MDA5 mutation demonstrate an interferon signature. But you can actually clearly differentiate non-symptomatic, asymptomatic, and symptomatic copper patients, copper mutation carriers, on the basis of their interferon status. So it's a bit like in dermatomyositis. You know, if you make a dermatomyositis patient better, the interferon goes away, and copper, it sort of has a feel of like that they're unwell when the interferon is high. But the weird thing about the MDA5s is that they've got rip-roaring interferon signatures, but some of them are clinically completely fine, you know, which maybe says that interferon is nothing to do with anything. I don't know. But so I think it's fascinating. You know, the non-penetrance is really interesting. And it's really in the dominant families that you get a feel for that. I would also say, though, you know, in a recessive family, you very rarely go testing asymptomatic children for their mutation status. And I don't think we know how much non-penetrance there is in recessive AGS, for example. That's my feeling.
I would just like to add, Yannick, thank you for this comment. You know, where I have the problem is when I find, for example, a novel mutation in the non-hotspot, you know, domain. And then I don't know what to do. You know, you find the novel mutation is predicted damaging, and then, you know, you keep thinking about, you know, reduced penetrance. You know, you find it in the parent. What does it mean? And, you know, maybe that mutation is found in a different domain, and as such may have a different phenotype than what is typically seen in patients. So now I'm thinking more about, you know, different phenotypes which are linked to mutations in the same gene. It's just that these mutations reside in different domains. So, you know, that is complicated.
Yeah, I mean, there's been a paper published, and we're trying to publish a paper, I think, with a JIA type phenotype, but with copper mutations. But they still have the signature.
Mm-hmm. Yannick, I have a question for you. Do you think there could be, because of your recent findings, there could be SNPs in STING that could somehow modulate this penetrance?
Yes. I mean, I don't, and yet I don't know. I mean, I think the work that Raffaella did with the digenic inheritance in the proteasome components is very interesting and very unusual, actually. But why not? Then I'm sure, you know, if it's happened once, kind of thing, there must be other examples, and you would think that those kinds of possibilities might exist. I mean, you know, STING and copper are not the same, but they do overlap. They can overlap. So, yeah, I mean, that's intriguing. And I think, I mean, more generally, I think that these polymorphisms in STING and in copper, why not, or MDA5, you know, they might predispose to adult onset lupus or this kind of thing. Yeah.
Thank you very much. I don't know if there are any other questions from the audience. Not so far.
There's a question from Mika for Ivona.
I think I answered the question, you know.
Sorry.
Yeah, so if you want, I mean, you can read my answer. So basically, or I will read Mika's question.
So the question was, again, regarding if you search for a virus cell death type in this disease.
Yes, that's what we are doing now. You know, these patients are rare, you know, so it's kind of hard to get cells from them. I mean, there is only one sharpin deficient patient in the world, that I can tell you that I've been looking all over the world for the second patient, yeah. And then, you know, so is with patients with HOIP deficiency, 2 of them, 1 of them died. We have another one. And then, you know, patients with HOIL deficiency also died. So basically, we are now collaborating with a group from the Rockefeller Institute, you know, and getting fibroblasts subset of patients with Hoyle deficiency and POLIP deficiency and Sharpin deficiency, and then we are going to look at into cell death pathways in all these diseases. But I'm sure that there is a major contribution of cell death pathways to the inflammatory phenotype in these diseases. And that's not going to be just the case with LUBAC deficiencies. I think that these pathways are understudied. We are not looking enough into cell death pathways in all patients with autoinflammatory diseases, whether we talk about TRAPS, whether we talk about, other, autoinflammatory diseases. Problem is that these experiments are not easy to do, so especially when you work in primary cells. So that's a limiting factor, but that's what we are trying now to do.
Okay, thank you very much again for all the speakers for this beautiful session, and thank you for the audience for the question. And I hope you enjoy the rest of the meeting.
Thank you. Bye.
Thank you. Bye.
Thank you. Bye.